In this video, we are going to learn about genetic code, some different perspective about this genetic code. So, genes act and express themselves by governing the synthesis of specific proteins. The functional property of a protein is determined by the sequence of its amino acid residues, which in turn is determined by the nucleotide sequence of the genes which govern its synthesis. The biological information essential for protein synthesis is provided by an information coding system called genetic code. It is located in DNA and mRNA in the form of nucleotide sequence. So, genetic code is simply the precisely coded genetic message or information stored in DNA and mRNA in the form of nucleotide sequences. It specifies the amino acid sequence of proteins. In brief, genetic code is the nucleotide sequence of DNA and mRNA that specifies the amino acid sequence of proteins. So, during protein synthesis, genetic code is first copied down from DNA to mRNA. This is called genetic transcription. In the next stage, it is transferred from mRNA to protein. This is called genetic translation. During transcription, the deoxyribonucleotide sequence of DNA is transferred to mRNA in the form of ribonucleotide sequence. During translation, the ribonucleotide sequence of mRNA is converted or translated to the amino acid sequence of protein. The idea of genetic code was first advanced by Alexander de Souza. However, it was further elaborated theoretically, and the first formal model of genetic code was presented by George Gamow in 1945 to 1954. Although Gamow's model was not accepted, it could serve as a pathfinder to open new avenues of thinking. The basic feature of Gamow's coding system that are still valid are the following. Genetic code is degenerate. An amino acid is coded collectively by a few codons. Codons are the basic units of genetic code. They are formed of bases and represent the amino acids of proteins. Codes and amino acids are collinear, that is, the amino acids in a polypeptide chain are the same sequence as the corresponding codons in DNA or mRNA. Currently, genetic code is visualized as a complex coding system composed of a series of coding units called codons. A codon is a nucleotide triplet. Formed of a set of 3 nucleotides. Therefore, it is often called a triplet codon. It codes for a specific amino acid. The sum total of the codons required for the synthesis of a polypeptide chain or a protein molecule is known as histone. The DNA molecule may contain many such histones. The 4 kinds of nucleotides of DNA or mRNA can enter into triplet combinations and form as many as 64, like 4 raised to 3 kinds of codons. These codes only for 20 kinds of proteogenic amino acids. So it becomes clear that some codons never code for amino acids, and at the same time some amino acids are coded by more than one codon. Those codons which code for amino acids are known as sense codons. Those which do not code for amino acids were formerly called nonsense codons. But they have been shown to signal the termination of a protein synthesis. So they are now more aptly called termination codons or stop codons. Of the 64 codons, 61 are sense codons and the remaining 3 are termination codons. UAA, UAG and UGA are the termination codons of mRNA. Some sense codons initiate protein synthesis or translation, so they are generally called initiation codons, like AUG in eukaryotes and AUG and GUG in bacteria. Then initiation codons. The termination codons are collectively called signal codons with distinction with sense codons. The sense codons which act together in groups or collectively code for the same amino acid are known as degenerate codons. We know that there are 61 sense codons, 3 remaining as termination codons. So Some sense codons which act together in groups and collectively code for the same amino acid are known as degenerate codons or synonymous codons or redundant codons. Of the known 20 proteogenic amino acids, only methionine and tryptophan are coded by the single codons AUG and UGG respectively. All others are coded by synonymous codons or redundant codons. 9 of them are coded by 2 codons each, lysine, cysteine, etc. One is coded by 3 codons like isoleucine, 5 by 4 codons each like valine, alanine, etc. and 3 by 6 codons each, leucine, serine and arginine. So we know that there are some degenerate codons or synonymous codons or redundant codons associated with this important genetic code. Then features of the genetic code. Now we are going to learn about features. Genetic code is formed of trinucleotide combinations called triplet codons. We know that the functional units of genetic code are codons. Each codon is formed of a set of 3 nucleotides and it specifies a particular amino acid. DNA and mRNA contain 4 kinds of nucleotides to code for the 20 proteogenic amino acids. So the genetic code is formed of trinucleotide combinations called triplet codons. The functional units of genetic code are codons. Each codon is formed of a set of 3 nucleotides and it specifies a particular amino acid. DNA and mRNA contain 4 kinds of nucleotides to code for the 20 proteogenic amino acids. If codons are presumed to be singlets, the 4 types of nucleotides can only form 4 codons, like 4 raised to 1, which means 4 codons, which in turn can code for only 4 amino acids. If codons are visualized as doublets, the 4 types of nucleotides can form as many as 16 codons, which can code for only 16 amino acids. However, if the codons are considered to be triplets, there would be as many as 64, like 4 raised to 3 codons, which are more than enough to code for the 20 proteogenic amino acids and also to provide start and stop signals in protein synthesis. Thus, it becomes all the more explicit that genetic code consists of triplet codons and each codon is a trinucleotide sequence. Next major point or feature, genetic code is universal. Until 1979, genetic code was considered universal. This means that the same code is present in all organisms. Each codon specifies the same amino acid in all organisms, from the simplest virus down to the most complex plants and animals, and the same signal codons have the same function in the vast majority of organisms. The universality of the genetic code was first received by Marshall, Cascade and Nirenberg in 1967. It probably reflects 3 evolutionary aspects as follows. Gene code got established at a very early stage in evolution. Genetic code is highly conserved in the sense that it has undergone little or no change in a long evolutionary history of life, although genomes have radically changed in different species and organisms have evolved from a very primitive to a most advanced level. Next, all forms of life have a common origin. Exceptions to the universality of the genetic code may be found in some prokaryotes, ciliate protozoans and mitochondrial genomes. These deviations are related to the termination codons. In ciliates, the mRNA codons UAA and UAG are not stop codons but are sense codons which specify glutamine. Similarly, UGA is not a termination codon in mitochondrial mRNA, but it codes for tryptophan, which in other cases is specified by UGG. Some alterations or exceptions to the universality of genetic code, like some animal mitochondria, Drosophila mitochondria, most animal mitochondria, vertebrate mitochondria, some animal and yeast mitochondria, some prokaryotes, plant mitochondria, yeast mitochondria, nucleus of Candida cyclindaceae, some prokaryotes, nucleus of some ciliate protists like Tetrahymena. Then some mycoplasmas and animal and yeast mitochondria, there are some differences. Next topic or another important feature. is genetic code is commaless. Genetic code is commaless. All available evidence suggests that genetic code is commaless. This means that it typically consists of a continuous series of codons with no information or indication at all to specify where one codon ends and other one begins. In other words, demarcation boundary signals are absent between adjacent codons. So all the bases in a polynucleotide chain are parts of different codons and none of them serves as a boundary between neighboring codons. Hence, deletion of a single base in all probability can cause a shift in the reading frame during translation. This may result in a protein with a radically different amino acid sequence. Then genetic code is unambiguous. Genetic code is unambiguous in the sense that a codon always specifies the same amino acids in all organisms. Also, it will not code for more than one amino acid all by itself, even though some amino acids are coded by more than one codon. Then genetic code has polarity. Genetic code is polarized with definite initiation and termination codons and a fixed or a definite direction for reading a message. This enables a gene to specify the same protein always. Also, it causes the code to read in a fixed direction. AUG, that codes for methionine, acts as an initiation codon. Therefore, the first amino acid to be incorporated in the synthesis of all proteins is methionine. UAG, UAA and UGA act as termination codons. Reading a codon in the opposite direction will specify another amino acid due to the reversal of base sequence. Then genetic code is degenerate or redundant. Genetic code is considered degenerate because one amino acid may be specified by more than one codon. The collaboration of more than one codon to specify an amino acid is called code degeneracy or code redundancy. Sense codons, with the exception of AUG and UGG, which code for methionine and tryptophan respectively, code for amino acids not individually but in groups. Thus, out of 20 proteogenic amino acids, 18 are coded by multiple codons. Only methionine and tryptophan are coded by single codons. The degeneracy or redundancy of the codon is believed to be due to presence of more than one kind of tRNA for every amino acid and the ability of the anticodon of a tRNA to bind with the codons of different kinds of mRNAs. Then genetic code is non-overlapping. The nucleotides of a codon do not normally overlap the nucleotides of an adjacent codon. This is not a universal phenomenon because there are instances in which adjacent codons do overlap so that the same nucleotides can serve as a part of two or more codons, like overlapping genome. Then genetic code has definite start and stop signals. Genetic code contains specific start and stop signals for protein synthesis. In all organisms, the mRNA codon AUG serves as an initiation codon and it codes for methionine, AUG for methionine. Then mRNA codons UAA, UAG and UGA do not code for amino acids but instead serve as chain termination codons or stop codons in genetic translation. Next topic, codon assignment or deciphering or cracking of the genetic code. Codon assignment or deciphering of genetic code is the process which determines the exact amino acid specified by a particular sense codon. In other words, it is the assignment of allocation of a specific amino acid for a particular codon. It was the establishment of the fact that mRNA forms the information intermediate between gene and protein which paved the way for the understanding of codon assignment. Then next topic, contributions of Nirenberg. In the mid-1950s, the triplet nature of the codon was almost revealed. The assignment of the allocation of a specific amino acid for a particular codon will be there. However, it was not quite clear as to which amino acid does a particular codon specify. The first breakthrough of the solution of this problem was made by the in vitro synthesis of a polypeptide by Marshall W. Nirenberg and his team in 1961 using artificially synthesized mRNA. The use of artificially synthesized mRNA is advantageous in that its base sequence is already known. Nirenberg and his associates completed the work in two stages. In the first stage, Nirenberg Severo Ochoa and their colleagues made long artificial mRNAs and determined which amino acid these mRNAs could incorporate into proteins. In the second stage, specific mRNA codons were synthesized and determined the amino-acyl tRNA amino acid tRNA complex to which each of them could bind. The artificial synthesis of long-chain mRNAs were made easy by the discovery of the enzyme polynucleotide phosphorylase by Gruenberg, Manago, and Ochoa in 1955. This enzyme differs from RNA polymerase in that it does not require DNA either as a template or as a primer for synthesizing RNA. In artificial systems, this enzyme can polymerize diphosphate nucleotides, forming a long single-stranded polynucleotide. The enzyme is found in all bacteria where it most likely serves as an exonuclease to degrade mRNA. So in 1961, Nirenberg and Heinrich Mattei added artificially synthesized mRNA polynucleotides of non-base sequence to a cell-free protein synthesis system of Escherichia coli and studied the incorporation of amino acids into proteins. The cell-free protein synthesis system consists of ribosomes, tRNA, amino acids, enzymes, and it was produced by breaking down the cells of E. coli and then separating its protein synthesizing components by gentle centrifugation. In this process, the mRNA of E. coli gets mechanically and enzymatically degraded. The cell-free protein synthesizing system is advantageous in that it contains virtually all components except mRNA. To make up this deficit, artificially synthesized mRNA was added to it. The cell-free system has the disadvantage that it is short-living, like functional only for a few hours, and is relatively less efficient in translation. Then Nirenberg and his team artificially synthesized mRNA using all bases and the enzyme polynucleotide phosphorylase. They isolated this enzyme from Asobacter vinylandi or Macrococcus lysodecticus. Nirenberg and Mattei at first synthesized poly-U messenger RNA, the mRNA formed for only uracil nucleotides, and added it to the cell-free system of E. coli. The result was the production of polyphenylalanine. Thus, the first artificially established codon is UUU, and it codes for phenylalanine. Further, Nirenberg and his team produced poly-A, poly-C, and poly-G messenger RNA. By introducing them to cell-free protein synthesizing systems, they could prove that AAA codes for lysine, CCC for proline, and GGG for glycine. This marked a phenomenal event in the history of molecular biology, namely the deciphering or cracking of the genetic code or the assignment of codons for specific amino acids. Nirenberg and his associates could initially decipher 4 out of 64 codons using homopolymeric mRNAs. For cracking the remaining 60 codons, they used copolymeric artificial mRNAs synthesized using 2 or more kinds of bases. They could establish that if 2 different bases are used in equal proportion, an mRNA with 8 kinds of triplet codons would be produced, and if 3 different bases are used in equal proportion, 27 kinds of codons would be the result. By using artificial copolymeric mRNAs, Nirenberg could crack the codes for alanine, valine, leucine, isoleucine, glycine, proline, serine, arginine, cysteine, methionine, tryptophan, and glutamic acid. Following Nirenberg, Ochoa and his team carried out similar studies. The efforts of these two groups complemented each other with the result that the empirical formula of many codons could be revealed. However, the exact base sequence of each codon, with the exception of AAA, CCC, and GGG, could not be established. In 1964, Nirenberg and Leder developed the binding assay or binding technique to determine the exact base sequence of codons. They could find that a synthetic trinucleotide or mRNA codon, a ribosome, and an amino-acyl tRNA like tRNA with an attached amino acid can form a complex in the presence of high polarity magnesium chloride provided the selected codon codes for the same amino acid that is attached to the tRNA. It was also found that A nitrocellulose filter can block the passage of this codon ribosome amino acyl tRNA complex across it. On the basis of these observations, Nirenberg and his group could determine which mRNA codon codes for a particular amino acid. Then Nirenberg and Leda artificially synthesized all the 64 kinds of mRNA triplet codons and attached them to ribosomes. These codon ribosome complexes were then introduced to a mixture of different kinds of tRNAs, all proteogenic amino acids Mg++ ions, and the enzyme amino acyl tRNA synthetase. With the help of the enzyme, each tRNA got attached to a specific kind of amino acid and formed an amino acyl tRNA complex. The anticodon of this tRNA then bound to a complementary codon of the codon ribosome complex. This codon ribosome AA tRNA complex were formed. The whole mixture was then passed through nitrocellulose membrane. The codon ribosome AA tRNA complex absorbed or adsorbed specifically on the membrane, and all other uncomplexed constituents freely passed through the membrane. The adsorbed complexes were then isolated and the amino acid and codon of each of them were identified. The base sequence of the codon of each complex codes for the amino acid of the complex. Thus, Nirenberg and Leder would determine the exact base sequence of the codons of most of the proteogenic amino acids. Then, contributions of Hargobind Khurana, we know him. Hargobind Khurana and his team, almost simultaneously with Nirenberg and Amatai, devised an indigenous method of determining the base sequence of the codons using artificially synthesized DNA and mRNA of known base sequence. Using synthetic DNA, Khurana and his team succeeded in synthesizing copolymeric mRNAs of known repeating sequences. This copolymeric mRNA were then used for genetic translation. The collinearity between the base sequence of the triplet codons of the mRNA and amino acid sequence of the resulting polypeptide chain gives a clear picture about the exact base sequence of a codon that specifies a particular amino acid. Initially, Khurana and his colleagues synthesized a copolymeric mRNA of the sequence. In it, the triplet codons UGU and GUG are alternatively repeated. The peptide chain formed by the genetic translation of this mRNA was found to consist of the amino acid cysteine and valine in an alternative repeating sequence. This reveals that UGU codes for cysteine and GUG codes for valine. By the same technique, using the different combinations of the four bases, Khurana and his team could determine the nucleotide sequence of the different codons which specify different amino acids. Thus, it becomes clear that painstaking effort Nirenberg, Khurana and his associates could bring to light the sequence of the codons which specify all proteogenic amino acids, and thus could compile the complete dictionary of the genetic code. They could establish that out of the 64 mRNA triplet codons, 61 are sense codons which specify amino acids. The remaining 3 are stop codons which signal the termination of protein synthesis and genetic translation. For this outstanding contribution, Nirenberg, Khurana and Hole shared the 1968 Nobel Prize in Medicine and Physiology. Then, wobble hypothesis. Wobble hypothesis is the proposition that the first 2 nucleotides of the anticodon of tRNA can pair only with the complementary nucleotides of a triplet codon of mRNA strictly in accordance with the Watson-Crick base pairing rules, whereas the third position needs not follow any base pairing rule. So it can bind with any nucleotide in the third position of different mRNA codons. In brief, the wobble hypothesis holds that codon-anticodon pairing follows stringent base pairing rules only at the first and second positions of the anticodon, and in the third position it exhibits some freedom or flexibility for unusual or non-complementary pairing without following base pairing rules. Wobble hypothesis was proposed by Francis Crick in 1966 to explain code degeneracy or redundancy of genetic code. According to it, only the first two bases of the anticodon of tRNA pair with the two corresponding bases of the triplet codon of mRNA as per the normal base pairing rules. The third base in the anticodon, however, has some amount of play or wobble that permits it to pair with any base in the third position of different mRNA codons. Thus, a single tRNA type can recognize and bind with two or more mRNA codons which different from each other only in the third base. For example, the anticodon UGC or UCG of serine tRNA can recognize two different mRNA codons like AGC and AGU. The binding between UGC and AGC follows the usual base pairing rule, but in UCG AGU pairing, hydrogen bonding takes place between G and U, which is a departure from the usual Watson-Crick base pairing rule. Such a type of non-standard or unusual base pairing is called wobble pairing. Wobble hypothesis also holds that a single tRNA cannot recognize more than 3 different mRNA codons, so at least 32 types of tRNAs are needed to decode the 61 sense codons excluding the stop codons. On the whole, it appears clear that the tRNA can recognize 1, 2 or 3 different mRNA codons depending upon its wobble base, the 5' anticodon base. If the wobble base is C, it will recognize only the codon that ends in C or G. G. If it is G, it will organize two codons ending in C or U. If it is U, it will recognize the codons encoding in A or G. The wobble base is never A because it would be converted to the base inosine, which would then pair with A or C or U. So that's all about this topics. Thank you for listening.